one of the aim of that study was to find some or to define prognostic markers or prognostic factors of treatment-free remission or stopping treatment, stopping TKI in CML. Because, well, we have already published a long time ago the first trials regarding TKI cessation, but now it's important to have also prognostic factors, and to do that we need many patients. And one of the goals of the Euroski was to include many patients, at least in Europe, from 11 different countries, to get enough events and patients to perform a strong statistical analysis. And the other aim or the other goal was also to reproduce what you have already reported in the different clinical trials, but also try or see a kind of evaluation of harmonized method of molecular monitoring. Well, the design of that study was to include patients, CML patients who have been treated with TKI at least 3 years, and include patients who were in a deep molecular response more than 1 year. And in that study, the definition of deep molecular response was what we say in our specialist vocabulary, MR4, molecular response at 4 log reduction of BCR-ABL. And in that situation, this was the criteria of stopping treatment. And then the patients were followed every month at the beginning, and then every 6 weeks with molecular biology investigation on the peripheral blood. And molecular recurrence, I mean the molecular relapse, was defined by loss of major molecular response. In that situation, we decided to rechallenge the treatment. Well, the key findings is that we were able to to reproduce what we have already reported on the different studies, such as TEAM study, but on a very large number of patients. In another word, more than 50% of patients are still in major molecular response after stopping treatment. And we reported preliminary results at 6 months, and we published these results at 6 months in Lancet Oncology a couple of years ago. And now we have performed the statistical analysis and the study after 36 months. So in another word, all of the patients, at least patients who have been included in the analysis, most of those patients have a minimum of follow-up of more than three years. Sorry, so 36 months, which was not the case as compared to the papers that we reported in Lancet Oncology. One of the clinical results of that study is that definitely we might be able to propose stopping treatment outside the clinical trials. And we have a little bit anticipated this point because we have reported last year, at least in Europe, the ELN recommendations regarding stopping TKI. So in the true life, it's possible to propose stopping treatment in patients who are in deep molecular response after TKI treatment. And and the other important implication is that it's important to continue to follow the patients a long time after stopping treatment, because we have noticed that we might be able to find some molecular recurrence after 6 months and after 36 months as well, probably. So it's absolutely necessary to continue to follow the patient who stop treatment long time after treatment cessation. One thing which is quite interesting and in progress is the fact that probably the molecular recurrence are really different when they are during the first 6 months as compared to after. In another word, probably during the first 6 months, the molecular recurrence are linked to the persistence of leukemic stem cells with a high level of proliferation. At least these leukemic stem cells are able to reinitiate the disease quite fast, as compared to the late relapse are more linked to something which is quite slow, which means that maybe the mechanism of molecular recurrence could be different. That could be, for instance, linked to the immune system or the immunology factors, which could be modified during that period of time, and that could be able an explanation of the late relapse. So clearly, we have a kind of two things which are quite different: the early relapse before 6 months and the late relapse after 6 months. And this is something that we are going to analyze now.